record on this computer. And it says recorded. Yay. Okay. Oh, you know what? I think I figured out how I did it wrong last time. It says record. It gives me actually got a menu to say where I want to record it. And if I ignored that, it didn't start recording. I bet you that's what I did to Keith. Keith is here. And Keith, I still feel terrible for missing your recording. But anyway, you can do it again. Sure. Okay. So here we go. I will start off with a brief introduction to our colleague, Dr. Brian Lanoil. Give a little bit of Brian's background, and then Brian can take things away. So Brian did his bachelor's degree at the UC Boulder, where he studied. He had a degree program was in molecular cell and developmental biology. And Brian, I wasn't able to figure out where you grew up. Are you from Colorado?
I grew up in Colorado, yeah.
Okay. Are you from Boulder or from Denver or from
Denver? Yeah. From
Denver. Okay, right, very good. After doing his first degree at UC Boulder, he did his PhD, again in molecular cell biology program at Oregon State, where he worked on, I guess, marine microbiology. That was the focus of your PhD. And then from there, he went to do a postdoctoral fellow at a really exotic sounding place. And I'd love to hear some stories about what kind of place this was like to work at, but at the NASA Jet Propulsion Laboratory at Caltech, where I guess you were a few years before the infamous Howard Wolowitz, Rajesh Kuthrapalli, Leonard Hofstadter, and Sheldon Cooper. They followed you a few years later.
They were my junior colleagues, I guess.
Very good. Although I think Sheldon might have had his PhD before you, but he had it at 16. Where Brian worked in this, I guess this is where your affinity for extreme environments came about. Is that right? You were working. You must have been working on the prospects for life on other planets or something like that. Is that right?
Yep.
Very good. So that's where Brian sort of cut his teeth on extreme environments. And following his stint at Caltech, he went to UC Riverside, where he had his first faculty position. He was an assistant professor there. And then he was recruited to the University of Alberta in 2008, and he arrived here as an associate professor. As I said, his research is focused on life in extreme environments, which feels very suitable today, given that we're having Arctic conditions outside. And Brian's going to tell us something about that, I think, about your work in extreme environments. And I understand, Brian, you've done work at both poles, so that's.
I have on bipolar, yes.
Yeah, very good. Okay, I'll let that one go. And with that, I'll hand the floor over to Brian. So please share your screen and entertain us for a little bit.
All right, let's hope that I can share this properly and it works as expected. So can everybody see my screen? Okay. All right, great. Okay, well, thanks, Dave, for that great introduction, and I appreciate the opportunity to talk to you guys about what I've been up to the last few years with looking at microorganisms and how they respond to a changing climate. This is one half of what my laboratory does. We look at a lot of things in Arctic systems and cold environments, extreme environments. I also work on looking at how we can use microorganisms as a tool for reclamation and dealing with hydrocarbon impacted systems. But I'm not really going to talk about that side of things today. I'm going to talk about the extreme environment side of things. So I titled this, How Do Microorganisms Respond to a Changing Climate? That's a pretty broad topic. So I'm going to tell you about several different projects that are going on in the lab, or have gone on in the lab. So I'll just start off by talking about microorganisms and climate change, because most of you are not microbiologists by training, although I'm sure that all of you are microbiologists by interest, at least to some extent, because microbes are really important for a lot of different processes in the world. They are involved in all kinds of nutrient cycling and biogeochemical cycling, and they produce and consume greenhouse gases. So as a result, they're strongly involved in the cycles that are involved in greenhouse gas production and consumption. And they also are strongly affected by their climate and the conditions in which they live. And so the microbial communities are both affected by climate change and affect climate change. And so as a result, there's been a lot of work that's been done on this, but not nearly enough, because we don't really understand very well how microorganisms respond to a changing climate, or how those changes in the microorganisms affect the feedbacks to the climate. And as a result of this, there was a paper published a couple of years ago by Richard Caviccioli and colleagues, including one of our own, Dr. Lisa Stein was one of the co-authors on this, which was a scientist's warning to humanity, microorganisms and climate change. And it was basically arguing that we don't know enough about how microorganisms respond to climate change and their role in climate change to be able to have a good understanding of climate change itself, because the microbes are so critical to this that this lack of understanding is actually driving our misunderstanding of of the system. So I'm not going to get into this too much other than to point out that that the microorganisms are important not only for the, processes in terrestrial systems, but also in aquatic systems, and actually even in clouds to some extent. And that things like things in the climate that are changing, including the direct ones such as temperature, but also the indirect ones such as changes in rainfall, changes in heat, and other things are going to strongly affect the microorganisms in the system. So I don't study all environments. My main focus is really on Arctic environments, and there's some reasons for that. And the main reason why I focus on the Arctic is because of this process called the Arctic Amplification. And the Arctic Amplification is the recognition that the Arctic is warming faster than anywhere else on the planet. There is some increase in temperature also at the South Pole, but most of the warming that's occurring is actually in the north. And you can see here on this diagram, which is coming from the NASA GIS system and via the National Sea Ice Data Center in the US, that the temperatures are at the North Pole are increasing as much as 5 degrees C, and even in the subarctic and high Arctic regions, it's between 2 and 4 degrees C as compared to temperatures that are the temperature shifts that are much lower in other parts of the world. And the reason for that, there's many reasons for this, but one of the main reasons is that sea ice is melting, and so that is actually changing the albedo of the Arctic Ocean. There's less reflection into space, more absorption of that heat, and so it's warming this region much faster. There's other reasons as well. I won't get into the details of that because you guys are all at least partially aware of that, I'm sure. So these systems in the Arctic are dominated by ice. They're glaciers, they're permafrost, they're sea ice. And as a result, because of that, not only are they experiencing the most change, but they're also the most vulnerable to change, because this ice can relatively easily be tipped over into a change of state and become water. And when that happens, these ice-dominated systems will change dramatically. So I'm going to tell you about stories from each of these environments. We're going to talk a little bit about glacier foreland soils first off, and then we'll talk about sea ice, and then we'll move over to permafrost. So this is from a paper that we published in 2016 with a couple of my former students there. Ido Hatam is now a professor at in Vancouver, and Sina Kazemi is now an environmental lawyer here in Edmonton. So what we're looking at here, this is the Duke River. This is a river that comes out of the Kluane National Park in southern Yukon. So this is a subarctic system, and this is the glacier at the head of the Duke River, which we call the Duke Glacier, even though it doesn't actually have a formal name, and it's probably would be named differently from the indigenous people who live there, but this is what we've been calling the Duke Glacier. And what we're looking at here is trying to look at the foreland soils here, so the area in front of the glacier. So the glacier goes up valley this way, and we're looking at this area in front of the glacier to look at how the microbial communities colonize these sites and undergo succession. And so why are we interested in this? Well, glaciers and ice sheets are retreating rapidly. So on the left-hand side of your screen, I hope it's on the left anyway, this side of your screen, we see the major ice sheets in Greenland and Antarctica. So these are continental ice sheets, are decreasing both in their overall mass as well as their retreating up valley. And what most of this is winding up in the ocean. On the left-hand side here is the sea level change as a result of this in millimeters. So we're already seeing several millimeters of sea ocean change just from the water that's coming off of Greenland. And Greenland, as you see here, is actually the one that's the ice sheet that's actually experiencing the most change. And that's because of this Arctic amplification that I told you about earlier. So we're seeing changes in ice sheets. We're also seeing changes in mountain glaciers. So the mountain glaciers are, these are all these different areas, 11 mountain regions around the world. And I'm not going to, this is a complex figure. I'm not going to get into this too much. This is data from the latest IPCC special report on the ocean and cryosphere and a changing climate. And the main point that I want to make here is that Western Canada and the US and Alaska both show very strong indications of decrease in the glaciers that are present in the mountains there. All 11 mountain regions show an overall decrease in glacial area as well as mass budget. And so these glaciers are retreating rapidly. And this is going to cause all kinds of downstream problems for people and for agriculture and for other things, but it also creates new opportunities. Which is that we create new soils in front of these glaciers. So what we're looking at here, this is at the Duke Glacier, the one that I was pointing out earlier behind me here, behind the figure here. And in 1950, this is an aerial photograph of this glacier in 1950, and you can see the full extent of the glacier goes down to about here. And then in 1957, it's retreated a bit. It's become thinner on the sides and moved up valley a little bit. And by 1979, it's moved up valley by quite a bit. And so this is the extent of the glacier here in the 1950 picture. And so we're looking at this and we're seeing this move up valley. And we were able to use aerial photographs like this, as well as a modern aerial photograph, in order to estimate the rate of retreat of this glacier and the age of the soils as a result by thinking of this as a chronosequence, a space for time substitution, where we're looking at the further away from the terminus of the glacier we are, the older we are. And so by looking at this, you can see that right at the terminus, we're looking at a few years. At the second site, we're looking at between 25 and 50 years. And then if we go all the way down to the end of our chronosequence, we see something that's about 260 years or so. And so by looking at this chronosequence, we're trying to see how the community establishes itself over time and whether we can see whether this is continuous change along this gradient or whether we're seeing different discrete communities appearing within this chronosequence. And what we see is the latter, that we see discrete communities appearing. So here are our youngest sites, the ones that have just within the last 50 years have been exposed. And we have two, an R and an L, and the R is from the right side of the glacier, and this is stage right. So if you're standing on the glacier, it's to the right-hand side, looking down valley, and L is on the other side of the valley. So we took parallel samples, and by doing that, we were able to see whether this was something that was just from a single chronosequence or whether this was clearly the result of some down valley trend. And so we saw that at the terminus of the glacier here, these guys that were right near the terminus of the glacier, that's one group. A second group is down valley here, and this is all the way from R5 or L5 all the way down to LR3, L or R2. And then once we move into the grassland, so this entire valley is devegetated. Basically, all we see here is mosses and lichens. We don't see any higher plants. It's not until we get down valley and into quite a ways that we get into the grasslands. And once we move into the grasslands, we see a very abrupt shift to a new community there. And so it looks like what's going on here is that we start out with conversion from a subglacial to a periglacial environment, and so that we get a community shift there. And what we're looking at here is the mean difference from the youngest site. So this is in comparison to site 7. So this is the mean difference of site 6 to site 7. And then we see a shift to a new community after about 100 years or so, or sometime between 50 and 100 years. And we see that this community is stable as long as there's no plants there, and so it doesn't really change. And then we see a final shift to a new community once we enter into this vegetated area. Now this may continue to shift as we go down valley. We didn't actually look at that. We were only looking at this, this transition zone. But it does look like we have this succession going from a newly exposed site down to a older exposed site to a site where we have plants in place. So it does look like we undergo bacterial succession. Other studies have been done that look like maybe protists and fungi also undergo succession. And this occurs, according to our study, prior to the plant succession. So Plant growth does have a significant effect on the microbial communities, but it looks like maybe these microbial communities actually prepare the soil for plant colonization. So the plants need the microbes to go in there, colonize it, and get it ready for the plants to come in. I mentioned that this is likely a secondary succession because we actually have microbes in the subglacial environment. So once they're exposed, they change to a new ecosystem rather than a new community, rather than it being a new colonization like it is with the plants. In the case of the plants, that's a primary succession. And in fact, there's been the classic successional study was done. One of the classic successional studies was done in Glacier Bay, looking at a chronosequence like this for plants. So why does all this matter? Why should we care about any of this? Well, glaciers currently cover about 11% of the Earth's land area. And as glaciers retreat, we are seeing new mineral soils exposed. We expect that most of those glaciers are going to be gone by, mountain glaciers anyway, are going to be gone by 2100. Now, only about 6% of the land area is mountain glaciers covered, as opposed to ice sheet covered. But nevertheless, we are still seeing significant areas of land that are going to be newly exposed, and we don't really understand how that process actually works. So we're trying to get a handle on how does the colonization work, and how do those microbes actually prepare the soils for the plants? And I don't have answers to those questions yet. Those are sort of the directions that this field is moving in. So that's, we just finished talking about glaciers and glacier forelands. And I've done work not only on the glacier forelands, we've also done work on the supraglacial environment, so the microbes that are on top of the glaciers, the end glacial environment, the microbes in the glaciers, and the subglacial environment, those that are below the glaciers. And we do see ecosystems within all of those places, and those ecosystems are also changing dramatically. But I don't want to spend our entire time talking about glaciers. I also want to talk about other environments in the Arctic. So I'm going to talk about sea ice extent. And we've heard a fair amount about this, or sea ice period. We've heard a fair amount about this kind of thing from Andy over the past several years, although he hasn't given one of these yet. So maybe Andy can do that one of these days. And what we see is that sea ice extent is decreasing dramatically. So the black line here is at the largest extent of ice. So this is in March, in the middle of winter. And the red line here is at the minimum extent of ice. That's in September. So at the end of summer, when as much is melted as is possible. And what we see is that the minimum extent of ice is dramatically decreasing, and the maximum extent of ice is decreasing somewhat. If we normalize this to 1995, this figure was done in the sea ice report card from NOAA. And so this is continuing onward. So we're seeing less and less extent of ice, but the other thing that we're seeing that you may not really be that aware of is that the ice is getting younger. And so what we're looking at here, this is 1985 up here on the top left. On the top right is 2000. So this was March of 2000. So it's the same time frame, March 12th to 18th. This is March 11th to 17th. So the same week basically. And what we see is that not only is the extent less in 2020, but the age is significantly less. And you can see that down here. There's far less 4-year-old, 3-year-old, 2-year-old, and 1 to 2-year-old ice, and lots more of this 0 to 1-year-old ice. So we're changing from this multi-year ice to this first-year ice. It's getting thinner and it's getting younger. So what are some of the features of these different kinds of ice that may actually be important here? Well, as I mentioned, the age and the thickness, but this also leads to things like there's a shallower thermal gradient in the multi-year ice because it's thicker ice, and the cold is on the top and the warmth is on the bottom, which is different than we're used to thinking, but the warm seawater is underneath and the cold air is above. And so there's a shallower thermal gradient for the multi-year ice than the first-year ice. There's lower salinity in multi-year ice because it's had a chance to drain through brine channels, and higher salinity in first-year ice. As a result, this multi-year ice has lower density and a rougher surface. There's less light penetration through this thick ice. And we form ponds on the surface of the multi-year ice, which we don't form on the surface of the first-year ice, because that first-year ice melts and disappears before the ponding actually can occur. Okay, so why does any of this matter, this shift from first-year ice or multi-year ice to first-year ice, and then the disappearance of the sea ice? Well, microbes are actually really important in these marine ecosystems. They actually drive all of the productivity because they're the photosynthesizers in the system, the algae, and they also drive all of the nutrient remineralization and much of the organic carbon processing in these systems. And so the sea ice is a big part of that in these environments. Microbes colonize underneath the ice, they colonize in ponds on the surface of the ice and snow, and they colonize within the ice within brine channels. And these microbes, as I mentioned, they're the primary producers. Here's the phytoplankton, and these phytoplankton produce organic matter that winds up in the dissolved organic matter, which is then consumed by the heterotrophic bacteria. And because these are then lysed by viruses, this goes back into the dissolved organic matter, and we have a loop that goes on here. And about 90% of the productivity, the biomass produced and the carbon produced and the energy produced by these phytoplankton is turned into either microbial biomass or returned back to its original form by the heterotrophic bacteria. They also are involved in pumping carbon to the bottom of the ocean through the biological carbon pump. I won't get into that too much. And as I mentioned, the multi-year ice has unique features that are not found in the first-year ice. And so it may have microbes that are unique to the multi-year ice that are being lost as we transfer it to first-year ice. So we did some work up at the Canadian Forces Station Alerts, the CFS Alert, which is the northernmost continuously occupied point in the world. It's, as you can see, it's on the northern tip of Ellesmere Island, and it's north of much of Greenland, and it's only about 500 kilometers from the North Pole. And this is a listening station. They use this in order to actually listen to other countries' broadcasts, military broadcasts, and try to interpret that information. And it's been around since the 1950s, and it's still used. This is what the station looks like from the air. It looks like just about any other military base. But they were nice enough to allow us to be there and to utilize their facilities in order to do the work. We had some limitations to us about where we could go, but we were able to use the facilities there. And if you go out this direction, we're looking at sea ice right here. So we were able to take skidoos and go straight from Alert out onto the ice. And this ice here is one of the few areas in the world where we still have both multi-year ice and first-year ice right next to each other. So we were able to collect samples of both types of ice right there. This work was done in collaboration with Christian Haas, who used to be over in EAS and is now at York University. And this is his former student, Ben Lang, who's now at the Max Planck Institute, and he's drilling a core of sea ice using our drill. And you can see he's got a drill bit in his hand, just a standard power drill with an attachment that goes onto this ice core. That's a custom-made ice core. And when you pull up the ice from doing this, the bottom of the ice, this is the part that was in contact with the seawater. This brown stain here, that's ice algae. That's phytoplankton that are growing on the bottom of the ice and undergoing photosynthesis. So despite the fact that this is snow covered and ice covered, we still have a high level of productivity going on within the ice. So this is just a map of the bulk salinity and the brine salinity of the ice. And what you can see here is that the bulk salinity is very low at the surface, and then it gets much higher as we go down. And this is because of that drainage or salinity that I talked about earlier. The brine salinity, actually, the salinity increases as you go up in the core. And that's because the brine concentration is determined by the temperature. So it gets thinner, the brine channels get thinner, but the salinity is determined entirely by temperature. And so the microbes are actually experiencing this level of salinity here. They also experience differences in pH as well as temperature within different parts of the ice. So we were thinking that maybe we would have different communities within the ice, and that is exactly what we saw. So first of all, if we look at the seawater, we see that the seawater is quite different from the ice. And so it looks like we have very different communities, microbial communities, living within the ice than live in the seawater. And also within the ice itself, we see at least three different communities here. This one right at the surface of 0 to 30 centimeters, this one here at the intermediate depths going down to about 150 centimeters, and these ones that go in the bottom from 150 to 200 centimeters. We don't know if that corresponds to years. It does appear that there's at least one annual layer within this ice, but it does appear that we have three different communities of microorganisms residing within this ice. And it's not just that we have different communities. These communities have different levels of diversity, and the diversity is quite high within the ice. We have different communities in the seawater, and yet we have diversity that's at least as high, if not higher, than what we see in the seawater. So this is a significant chunk of the biodiversity in the system overall. And here's our ice average using, this is two different diversity indices. I won't get into the differences between them too much here at all, just to say that we do see differences in diversity as we go down the ice, and that it's at least as high, or in many cases at least as high as the seawater is. So it looks like what we have going on here then is microbial communities are quite distinct within the ice. It's not just that we have these unique communities within the ice, but also when we look at comparing first-year ice and multi-year ice, we also see differences as well. And so we have multi-year ice as our squares here, first-year ice as our circles, and seawater as our triangles. And what we see is that first-year ice looks like, sometimes it looks like seawater, sometimes it looks like something that it's basically its own thing, and sometimes it looks like multi-year ice. And so we see a lot more variability within the microbial communities within the first-year ice than we do within either the multi-year ice or the seawater. And we also see that when we look at these measurements of diversity. These three are all different measurements of taxon richness, species richness, and we see that first-year ice is much more different kinds of taxa. And we also see that the overall diversity is also higher in the first-year ice than in the multi-year ice or seawater. And that's observed further when we look at the overlapping taxa in these communities here. So our first-year ice, these are OTUs, operational taxonomic units. You can think of these as sort of semi-equivalent to bacterial species. And we see that first-year ice has a significant amount of overlap with seawater. About almost 100 species are found in both first-year ice and seawater, to the exclusion of multi-year ice. There's a lot of overlap between first-year ice and multi-year ice, 125 species or OTUs, but there's very little overlap between multi-year ice and seawater. And we see only 16 that are found in both multi-year ice and seawater that are unique to those two communities. So it looks like what's happening here then is that first-year ice is somewhere in between. It's sort of a mix between multi-year ice and seawater, but multi-year ice and seawater are not very similar to each other. So the sea ice bacterial communities differ from the underlying seawater communities, and multi-year ice has a complex community structure at different depths, and it looks like we have different communities within different parts of the sea ice. First-year ice is more variable than multi-year ice, and so what does all that mean then? Well, we know that sea ice is becoming younger, which indicates that there's some loss of microbial communities from these systems, as well as increased variability, both of which have unknown consequences. We don't know what's going to happen to these systems with the loss of these communities. If the sea ice disappears entirely, then the entire microbial community that's unique to the sea ice is going to be lost from the marine Arctic system, again with unknown consequences. So the future directions of this is we need to better understand the consequences. We don't really know what happens when these communities are removed. What is the role that these microorganisms play in the ecosystem functioning in these environments, and what does their loss mean for these ecosystems? Okay, so now we're moving on to our last environment here, which is permafrost, and we're going to talk about a couple of different aspects of permafrost. We're going to start off by talking about permafrost as an archive. So we've talked about glaciers, which are retreating. We've talked about sea ice, which is disappearing. One thing that's happening with permafrost is thawing and melting. And so we're going to talk about that as our second aspect of things. But the other part of permafrost is that permafrost acts as an archive. So a lot of what we know about the past, past history of the planet, especially the more recent past history of the planet, the past several million years, we know from climate reconstructions from ice cores, as well as from permafrost. Permafrost is much younger, and so it's more recent. But we can tell a lot from looking at what's going on in the permafrost, because permafrost preserves signatures of past climate. And these can be chemical signatures like isotopes, or it can be biological signatures like dead animals and plants. So fossilized, not really fossilized, not siliconized, but silicified rather, but dead and preserved and intact. And so we can actually even extract DNA from these organisms. There's been a lot of work done on ancient DNA in permafrost. Less work done on glacier ice, but there's been some work done in that area. And by looking at these kinds of things, we've been able to reconstruct past climate and past ecosystems. So what we're looking at here is a timeline. And so on our x-axis here, we have years before present. So here's the present day, and here's going back up to 15,000 years. And the whole of human history is right here in this period from about 12,800 years ago until, 11,600 years ago until the present. That's when all of human history occurred. And that's all in this period we call the Holocene. And before that was the Pleistocene. And so this period here, this is called the Younger Dryas, which is a period where there was abrupt cooling and then a shift to abrupt warming. And when that happened, we shifted from a relatively steady state cold condition in the Pleistocene, this is globally, to a warmer condition during the Holocene. And so I have a picture here. Let's see. There we go. So this period here was a period of abrupt change. So you can see the temperature went from very cold to very warm in a very short period of time. This part right here was only about 50 years. So it warmed by about 10 degrees C globally within about 50 years. And then over the next several thousand years, it continued to warm by another 10 degrees. So we went up by 20 degrees C over that period of time. And then we've had some fluctuations. We've had a cooling here. We had the little ice age here, but nothing that was anywhere close to the temperatures that we saw during the Pleistocene period. Okay, so what this means is in the region that I'm working in here for this particular study, this is called Beringia. This is the area of the northern Yukon and Alaska and into Siberia that we call Beringia. And there was an ecosystem that was present there during the Pleistocene that included a lot of megafauna. This was mammoths, and there were horses, and there were things that were like rhinoceroses. There was also saber-toothed tigers and other kinds of things in these regions. Long-toothed tigers and things, large cats, that all went extinct during this transition to the Holocene. So various different animals went extinct. We also had novel organisms moving into these systems, as well as some organisms that survived. The novel organisms include things like moose and humans. The things that survived were things like caribou and muskox that are present in these systems as well. And so we had a massive change. There was extensive extinctions that occurred and extensive changes in these ecosystems that occurred along with this warming. So we did some work looking at, this is at a site off of the Dempster Highway in the Yukon, and we drilled the core there. We collected samples right at that Pleistocene-Holocene interface to try to see whether we could get a handle on how microbial communities changed at that time period. Now, when we look at within a time period, so if we look at within the Holocene or within the Pleistocene, there's been a few studies that have been done that show that microbes are not easily correlated to these changes. And that's in fact, the only thing we can see is that age seems to be an important factor in determining the composition of the microbial community. But until this study, nobody had ever looked at this transition between the Holocene and the Pleistocene. And as you can see, the environment is quite different. This is the material coming out of the Holocene samples and coming out of the Pleistocene. This is mostly peat, and this is mostly sand. And so not only was the environment quite different, but so were the microorganisms. So what we're looking at here is cell abundances, and we can see there's many more cells in the Holocene samples than in the Pleistocene samples. We can also see that more of the cells are alive in the Holocene samples than in the Pleistocene samples. So there's about 8 times more viable cells in the Holocene than the Pleistocene samples. There's about 2 times more dead cells in the Pleistocene samples than the Holocene samples. And finally, there's 2 times more total cells in the Holocene samples than the Pleistocene samples. So we definitely see a shift here. This could be because of age, or it could be because of these environmental factors that are going on here. And so we took a look at the environmental factors, and there's a whole list of them here that you can see. And what we found is that there was, within an epoch, there was very little variation in these environmental factors, and we couldn't use them to explain the distribution of our communities. So it didn't look like there was any correlation between chemistry or other environmental parameters and the communities within the Holocene or within the Pleistocene. So within an epoch, those samples didn't really vary in a way that we could really differentiate using chemistry. But between epochs, we saw that everything was actually significant. So everything was significantly different. There was a major shift in the soil chemistry, and that was accompanied by this major shift in the composition of the microbial community. So it looks like what's going on here then is that the microbial community structure within an epoch is quite stable over time. And we think that this is likely due to microbes' high physiological flexibility. So they're able to adapt to relatively minor changes in temperature or water content or things of that sort. But if the system surpasses some kind of ecological threshold and changes dramatically enough, you'll have an abrupt shift to a new steady state, at which point it will become stable again. So we are now going into a period that we're calling the Anthropocene. There's a shift from the Holocene to the Anthropocene, where we're seeing big effects from climate change due to human-driven causes, and that may lead us to this ecological threshold event. If something like that does occur, we will have an abrupt shift rather than a gradual shift in the microbial communities in these systems, is the most likely scenario based on this study. Okay, so let's continue then. We'll talk about these permafrost. So that was permafrost as an archive. Now we're going to talk about the effect of climate change on permafrost and on permafrost microbial communities. So permafrost is frozen soil that's been frozen for at least 2 years, and maybe as much as 1 million years. So we're looking at a wide range of ages of these things, but they're all ice-containing soils. And so what happens when you warm them, if you warm them enough, is they thaw. And when they thaw, you can get massive changes in your landscape. And this includes things like thermokarst pond formation, like what we see here. If you're on a flat land area, what happens is you get deflation because the water is takes up less space than the ice does. And so the land collapses and you end up forming these depressions across the landscape that are filled with water that is the result of the thaw of the permafrost. And so you get these thermokarst ponds that form. This is also a type of thermokarst formation. What we have here is a thaw slump where we're on a hillside and the permafrost thaws, and then we get a collapse of this hillside, and then this can often lead to erosion into nearby freshwater systems, rivers and streams. I do have a project with Suzanne Tank on this. I'm not going to be talking about that. Most of what I'm going to be talking about in this presentation is going to be a collaboration with Dwayne Fries over in EAS. But the point is that these abrupt changes to the environment are accompanied by very massive changes in the release of carbon. So a lot of carbon is released to the atmosphere when we do these abrupt changes. There's a lot of the Arctic terrestrial ecosystems are subject to this rapid thawing. Everything in red and kind of black or magenta up here is subject to rapid thaw. The stuff in blue here is subject to more gradual thaw. And so as these landscape changes occur, there's lots of carbon release, and this appears to be something that's going to be going on well into the future. The red lines here, the solid red lines, are the projected undisturbed permafrost or undisturbed tundra that remains. And you can see that we're on a decline on all three of these models, going well into 2300. We also see an increase in both the active thaw and the stabilized thaw that occurs in this. The details of these models don't really matter for the purposes of this presentation, but the point is that we're going to be seeing this thaw and the effects of this thaw well into the future. And so this is important because permafrost contains 2 times as much carbon as we have in the atmosphere. We also have about 50% of the world's carbon and only 15% of the land area, which is underlain by permafrost. And that's increased by 1.5 times if we include the deep carbon that's there below 3 meters. And so now instead of 50%, we're looking at closer to 75%. And we have over 1,000 petagram is 1 billion grams of degradable organic carbon that's present in these soils. So if that carbon is released from its frozen state into a thawed state and microorganisms can attack that, that carbon may wind up in the atmosphere, and that could lead to a positive feedback effect on the climate, a runaway positive feedback effect, where we have lots of carbon going to the atmosphere leading to more warming, which then leads to more carbon release. Thus far, that has not happened because increased plant growth as the result of increased warming has taken up the carbon that's been released from the soils. But we seem to be at a tipping point now where it looks like, if we take into account this abrupt thaw, like these soils, these permafrost soils in the Arctic look like they may be at the point where they may be becoming a source, or they may already have become a source rather than a sink for carbon. Okay, so what about the microorganisms? Well, if we look at the microorganisms in these systems, we know that when we thaw permafrost rapidly, we see activity. There's a rapid release of methane from the permafrost. That's mostly abiological, but we do see rapid consumption of the methane. And when the microbes do that, they consume that methane, and that doesn't escape to the atmosphere. We also see rapid shifts in the communities, which are the arrows here, especially here. So so this is in the composition of the community, and this is in the function of the community, or potential function of the community. And we see a very dramatic shift right away. On thaw, within 2 days, we see a shift in the community function, as well as some slight shifts in the community structure. Okay, so we know that microbial communities shift in response to permafrost thaw. We also know that their activity shifts. This is from a study that was done in my lab by a former graduate student of mine. And what we see is that with warming, we see a linear response to temperature in both permafrost soils, permafrost affected soils. We see a linear increase in methanotrophic activity. And if that soil is disturbed, we see a much more dramatic increase. So disturbance in this case was due to a bulldozer that came in and removed vegetation, but disturbance could also be a thaw slump or a thermokarst pond formation or other kinds of disturbances that may lead to increased activity of microorganisms in these systems. Okay. Also, long-term deep field warming experiments, this is not from my lab, this is from another lab, can lead to shifts in the bacterial community composition. And so what happens is that when you do the warming, it looks like the intermediate layer, this layer at the bottom of the active layer. The active layer is the soils that overlie the permafrost and that are actively frozen and thawed every year and that are more biologically active than the permafrost. The intermediate layer comes to resemble the active layer much more than the permafrost layer, whereas in the control, it's much more like the permafrost layer. So it looks like on thaw, the intermediate layer of soils resemble active layer soils. And we tried to get a handle on why that might be by looking at a site that was nearby Dawson City. This was an active mining site, so this is placer gold mining. So they're stripping away the permafrost and looking for gold in here. And what they did here was they removed the vegetation in order to start the mining process. And during that process, they within several months, we saw this gradient of disturbance here, going from this undisturbed site up here into a thaw pond down here. This is like a thermokarst pond down here, and we could move along this gradient and take samples along here. We took both cores, which had permafrost, as well as thawed intermediate layer and active layer in it, as well as the second core that only had intermediate layer and permafrost, and this one that only had permafrost. We also took surface samples along this gradient, and 3 of them match up to the position of these cores as well, so that we can match up the surface layer activity to or function to the cores. And so when we looked at these, we found something interesting, which is that this was within a few months of them having started this thaw. And when that occurred, there was no shift in the chemistry. We didn't see a change in the chemistry across these different sites, which indicates that the chemistry has not yet adjusted to the disturbance. But when we look at the microbes, they did adjust very rapidly. And we found that the permafrost was very dissimilar to the surface layer, the active layer. So here's our active layer, the top of the core 1, as well as sample A, which are both the active layer. And our permafrost is all looking over here. And what we see is that all of the disturbed sites and all of the intermediate layers in the cores all match up. So it looks like as soon as you thaw the permafrost, it starts to look like that intermediate layer, which is what was seen with that warming experiment, where we can see this within a very short time frame. And we wanted to know whether this was due to the permafrost community dying off and basically being non-viable, or whether they were still present, but they were invaded by the active layer community. And so we did an indicator species measurement, and we found that along this gradient, we always saw permafrost bacteria indicators within all of these samples, indicating that the permafrost community was still present. They were just overwhelmed by the abundance of the active layer community, which indicates that it's not due to species loss, but rather due to invasion. All right, so in sum then, our anthropogenically driven permafrost thaw did not lead to dramatic shifts in the soil chemistry, but did lead to major shifts in the bacterial community composition, which means that the bacterial community composition shifts actually precede the changes in the chemistry. So there's something else going on besides chemistry that's driving these microbial communities. The thawed permafrost resembles the active layer community, not the frozen community. And these indicator species remain. So that taken together means that the thawed permafrost looks like it's rapidly colonized by nearby active soils. So what does that mean? Well, if that holds true everywhere, which, obviously this is a single study at a single site, that may indicate that permafrost bacterial communities don't actually play a significant role in soil microbial processes, but rather active layer communities play a significant role. So future studies then maybe should including greenhouse gas fluxes, should focus on the active layer soil communities, maybe with the permafrost chemistry. So the organic matter from the permafrost thawing out, but then the active layer communities attacking that permafrost carbon. So that's the implications of that study. So overall for permafrost then, the microbial community composition in soils is quite stable and able to adjust to relatively small scale shifts in the environmental parameters, but they can change when a threshold is exceeded. Also, permafrost microorganisms are not as important for consumption of carbon as active layer microbial communities, and disturbance can affect microbial community activity as much as temperature change can. So abrupt permafrost thaw could have wide-ranging implications for soil bacterial structure and function. Okay, so I've told you now about glaciers and sea ice and permafrost, so all these different environments that we're working on and microbes that are there. So what is sort of the big picture overview then? So microorganisms appear to be responsive to disturbance and climate change, so we could use these as a way to integratively look at markers of climate change impacts on ecosystems, and I'll tell you about my idea with that in a second. Second, microbial responses to climate change in the Arctic and subarctic are complex and they require more study. You can't just make a prediction based on what we know now. The loss of microbial communities resulting from climate change could have unknown but important implications for ecosystem functioning. So this allows us to obtain, we need to obtain a better understanding of how shifts in the microbial community structure impact microbial community function, and that's the direction we're heading with this. And also, these cold environments are really great systems for understanding microbial ecology and climate change impacts. That these Arctic systems in particular, which are being strongly affected, allow us to get handles on things that are much more subtle at lower latitudes. Okay, I mentioned that I would talk about using microbes as markers of disturbance, and I'm just going to give you sort of the general gist of this. We're doing this a lot more in our oil sands work than we are in the climate change work, but the concept applies just as much, which is that we have a range of natural variability or a baseline that we're starting with. And we want to look at how far away a disturbed or changed site is from that range of natural variability. And we can use bacterial community composition or community function or the level of variability within those in order to get a handle on that. And if we follow that over time, we can see whether the communities are becoming more or less disturbed, and we can see whether they're moving back toward this range of natural variability, or for example, in this picture here, if a soil was looking like this boreal forest soil over here, and now maybe is underneath this pond, is it starting to look more like Arctic freshwater ponding systems than it is like the forest soil systems, and how long is it going to take before the community comes to resemble that of other natural Arctic pond systems? That's one way of looking at it. We can look at the movement of this group toward a reference community. We can also look at how the variability of that community changes. Does it decrease or increase? And so here we have one that's moving toward a particular community. Here we have one that's moving away. And so by looking at these things, we can quantify how far away we are. We can also look over, if we're watching over time and seeing how it's moving, we can talk about trajectories and how long it's going to take for this the system to move into a new state. We can talk about rates. We can talk about other aspects of things which we can't get access to now, not in as integrative of a way as we can with the microbial community. So we're proposing to start working on this, or in climate change systems, as much as we've been working on this in oil sands and other hydrocarbon impacted systems. With that, I'd like to acknowledge my lab members. So most of what I talked about is things that were in either recent or not so far distant theses. So Dr. Alireza Saidi Magraba just finished his PhD in December. He's now going to a postdoc at the Desert Research Institute in Las Vegas. Patrick Newberger is a former master's student who's now doing his PhD at McMaster's. Sina Kazemi is a former master's student who's now an environmental lawyer here in Edmonton. Ido Hatam is now a professor in Vancouver, and Tanya Strelitz is a former undergrad student who is now at the University of Texas doing a PhD. So with that, I'd like to see if there's any questions, and thank you.
So Brian, if you don't mind, I'll kick off the questions.
Right.
Can you comment on the species that you think are being lost? And are those the ones that are sort of more temperature sensitive, so to speak? Or are there other things going on with the loss of the species that you're seeing in the changes?
Right, so we don't see any real correlation to, well, okay, so first of all, let me just say, when the approaches that we're using, these DNA-based approaches, don't really give us a great handle on the traits of the communities, because we have to look more closely at their metagenomes in order to really get a handle on that. We've mostly been doing compositional measurements here, looking at ribosomal RNA-based associations. And so we can't really say for sure that these are cold adapted or warm adapted organisms. So we don't know for certain whether these are, for example, capable of surviving at warmer temperatures. What we do know is that they are unique to the sea ice or the glacier or whatever communities. And so by removing those environments, we're removing those communities from the system. So we don't actually have a great handle yet on what that means for these ecosystems. So the species that we're seeing are not particularly unique in terms of, they're not all that different from things that you would see in terms of, they're not further distant from their nearest neighbor than you would see in seawater, for example. So unlike maybe in certain systems, like in hyperthermophilic systems, we don't see that there's sort of this unique microbial community in the cold environments. What we do see is that at the species level or the strain level, maybe those functions may be lost from the system, or the communities may be lost from the system.
And maybe just a quick follow-up question. So the microbial diversity that you see in, say, Arctic seawater, is it different than what you would see in the more temperate seawater?
Surprisingly, no. So one of the things that there's been a big push to sort of bring a lot of these concepts from ecological theory into microbiology over the last 20 years or so. And one of the things we don't see is we don't see a strong latitudinal gradient. So we don't see that the number of species decreases as we go to a higher latitude. And so there doesn't seem to be a correlation there. And in fact, it's more environmental factors that seem to control the microbial community composition. So pH is one of the main drivers, for example, of microbial community composition, much more so than where we are on the planet.
Okay, great. Thanks very much, Brian. Great talk.
Thanks, Warren. Rolf?
Hey, Brian. Great talk. I'm just wondering, you kind of like, we deal with different kinds of organisms, but we have the same sort of multivariate approach with all the ordinations that you were showing.
Sure.
So I was just wondering, I mean, you show a number of ordinations based off of taxonomic diversity, and then you did show some also based off of, I assume, functional traits, right?
Most of the ones we were looking at were chemistry, actually, not so much functional traits. So ecosystem traits.
Yeah, but yeah, right. So you had some ecosystem traits. I'm just wondering if have you ever kind of compared the amount of variance that you find in the taxonomic analyses using the ordinations versus the more like functional based approach, and if there's some sort of indication that even though you might get very, very pronounced changes and shifts in community composition, the underlying functional redundancy is so great that the functional shift doesn't really seem to occur as much.
Right. So not so much in the systems that we've been studying. We haven't got a good handle on that quite yet. But what we do see is in other systems, in other studies that have been done, they do see some level of functional redundancy. So there is definitely a high level of functional redundancy in microbial systems because we have a very high diversity within those systems, high richness. Yeah. And so a lot of organisms can kind of step into that role and take over if there's a loss of a community member. And so there's definitely quite a bit of that going on there. However, what we still see is we still see shifts in the functionality of the systems. And so that's what I was kind of saying to some extent with the permafrost as an archive, is that there the communities are fairly stable, but also the functions are quite stable within those communities. But once you reach a threshold point, and then you rapidly change to a new trait-based system as well.
Right. Thanks, Brian.
Thank you.
Brian, it's Aaron. Again, very nice talk. What are some of the dispersal mechanisms that would allow the system to shift? I mean, you see rapid shifts, so obviously some of the things are just there, right? And they're just becoming more or less dominant. But do you think you see whatever long distance dispersal is in microbial communities? But how does it work? I mean, what are some of the mechanisms?
Right. So there's an old saying in microbiology by this Dutch microbiologist named Bas Becking, who said, "Everything is everywhere the environment selects." And it's not really very clear at this point whether there is such a thing as a biogeography of microorganisms, whether there is some kind of restriction on their dispersal, because they're so small that they're very easily carried by wind and water and other mechanisms. So it's very hard to create some kind of physical barrier. And so we often see evidence of sympatric speciation going on in these organisms because there is no real barrier between them. If you go to any pond around the world, if the chemistry is the same, you're going to find the same organisms there. And so the selection is really based upon the environmental parameters rather than dispersal mechanisms. And so there doesn't seem to be that kind of restriction. You'll find the same organisms at the South Pole as you find at the North Pole. So if you go to the Arctic marine systems, you'll see the same thing. And that may be a little bit surprising, and certainly for animal and plant folks, that's like heresy to some extent, because I know that's like a big driving factor in how you actually do things in these systems. But in microbial systems, it's really not, doesn't seem to be a major restriction on how how the ecology actually works.
Thanks.
Any other questions? I have one, Brian, and I'm, you talked a little bit about this, but the consequences of these shifts in composition. So for example, when you go from old ice to younger ice to seawater, and you see these great shifts in composition, what does it mean for function? I mean, how do you get at that? I mean, can you look at the species composition, or I guess it's not even species, is it? It's a higher level diversity index than that. And is there a way to model what that means ecologically or environmentally?
So, I mean, one of the things is sort of what Rolf was talking about, to look at trait-based mapping and trying to figure out what these organisms are doing. And that can be done pretty well in systems that we know really well, like the human gut, for example. So you can map traits to particular organisms within the human gut and do a trait-based analysis that way. But really the best way to do it with microbial communities is to use a metagenomic approach, where we're actually looking at all of the genes and all of the organisms and within a system, and using that as a genetic potential, and seeing whether we're losing particular features of the community as we lose members of the community. And so that means mapping those features to the members of the community, which is not so easy to do, but that's definitely the direction the field is moving, is to use genomes from all the organisms in the system and just sequence everything there, and try to then figure out whether the shift actually has a significant change in the ecosystem function. I mentioned this a little bit when I talked about the thawing of the permafrost in the laboratory, that they saw a shift in the function, And that was due to the genes that they could find within those communities. Those shifted dramatically, both in their abundance, but also in their representation, whether they're present or not in the community. And then if we can figure out what those genes actually do, then we can say what that loss might be.
Right, and then when you're dealing with these sea ice ecosystems, of course, there's other players in it, right? In the plankton, and you have no idea how those interactions work when you take it into a laboratory. So it gets complex in a hurry, I guess.
It does, and there's been quite a bit of work done on looking at these interactions, sort of transfer of carbon between different players and things of that sort. But that's all at sort of a systems level kind of thing, where it's, you know, carbon moves or energy moves from one player to another, one group to another, where they put them into boxes, and we're trying to kind of pull apart those boxes a little bit and see what's going on inside.
Okay, thanks, Brian.
Thank you.
Do we have any more questions for Brian? I don't see any blue hands, and I don't see any visual hands. So if that's the case, Brian, I really want to thank you. That was fascinating. Very cool stuff. Lots of food for thought. And the other thing I should do is remind everyone that next month on March 11th, we'll have Jacob Berry. We'll be giving the next chair's lecture, so we're looking forward to that. And the final comment to you, Brian, and I really want to thank you for doing this and for doing such a great job, is that you will find in your mailbox sometime soon one of these highly coveted Department of Biological Sciences inspired discovery for a better world water bottles. Get them while they last. So congratulations, Brian, you've earned it. And that was really great. I really enjoyed it.
Excellent motivation for others to do the same.
Exactly. So if there's no further questions, I want to wish everybody a good day, and keep warm in those extreme environments out there.
Absolutely.
Okay. Thanks, Brian. Take care.